Well, good afternoon. Welcome to the 2020's first Neuromodulation Journal Club. Next slide, please. Before we begin, as always, I would like to thank our engineering and science program committees for their continued support of our monthly initiative, and my colleague, Dr. Irina Dav, for her dedication and commitment. And of course, many thanks to our coordinators for facilitating today's event. Next slide, please. Well, for those who is joining us for the first time, I would like to share the brief mission of the Journal Club. We have been directing the Neuromodulation Journal Club since 2020, and our main goal is to bring together the neuromodulation experts and experts from the neuroscience, engineering, and clinical fields with interest in neuromodulation, provide a platform for trainees to present their newly published scientific papers, showcase the technical advances of each research group from diverse disciplines, and discuss the various aspects of neuromodulation research. And we hope these meetings will become an excellent platform for new exciting collaborations among scientists, engineers, and clinicians. Next slide, please. We have several ground rules. This is a 1-hour webinar. We will have 45 minutes presentation and 15-minute discussion at the end. You will be in the listening mode throughout the presentation. You can type your questions or comments to the chat or Q&A box anytime during the talk. We will address them at the end. If you have experts today in the field among us, please feel free to share your thoughts, experiences, comments. We appreciate your contribution. And last but not least, the webinar will be recorded, so you will be able to access to it on NAN's website. Next slide, please. I don't have any disclosures relevant to today's talk. Next slide, please. Well, the title of this month's paper is Long-Term Ecological Assessment of Intracranial Electrophysiology Synchronized to Behavioral Markers in Obsessive-Compulsive Disorder. And today's speaker is Dr. Nicole Provenza. Dr. Provenza is a postdoctoral fellow in Dr. Samir Shet's lab at Baylor College of Medicine. She completed her PhD in biomedical engineering in Dr. David Barton's lab at Brown University in 2021, where she was awarded the Draper Fellowship for her graduate research. Her graduate work focused on developing adaptive deep brain stimulation for obsessive-compulsive disorder. As part of her graduate work, Nicole developed a research platform to collect intracranial local field potentials synchronized with disease-relevant behavior at home to aid in identification of neural biomarkers of obsessive-compulsive symptoms in natural environments. Well, without further ado, Nicole, take it away.
Thank you so much for that introduction, and thank you so much for inviting me. I'm really excited to be giving this talk here. So I am a postdoc in Samir Shet's lab, the Functional and Cognitive Neurophysiology lab, and our group studies human decision-making and cognition, and we develop novel therapies for psychiatric disorders. And there are 3 main techniques we use to do neural recordings, and those are intracranial recordings in the epilepsy monitoring unit, including local field potential recordings and single unit recordings. And we do intracranial recordings in patients with DBS systems that are capable of concurrent sensing and stimulation. And we're involved in 2 clinical trials currently. One focuses on the personalization and optimization of DBS for individuals with treatment-resistant depression. And in this trial, DBS leads are implanted in the DBS targets along with temporary stereo EEG depth electrodes in limbic networks. And they stay in the hospital for 9 days and do extensive behavioral testing in our EMU, the epilepsy monitoring unit. And we use this data to optimize DBS parameters and simulation fields. So that's where these networks that are highlighted in this figure are showing. And so after this monitoring period is over, the stereo EEG electrodes are removed, but the DBS electrodes are meant to be permanent. And then the second clinical trial that the lab is involved in is developing adaptive deep brain stimulation for obsessive-compulsive disorder. So that's where these 2 figures are coming from, and that's the work I'll be talking about today. And so this is our lab, and first pictured is our PI, Samir Shet. There are several postdocs, a neurosurgery fellow, 2 research coordinators, a PhD student, and 2 medical students. So So the overarching goal of this work and my work in general is to improve the efficacy of deep brain stimulation for refractory psychiatric disorders. And in this work, obsessive-compulsive disorder. So OCD is a psychiatric illness marked by obsessions, which are recurrent distressing thoughts, and compulsions, which are repetitive ritualistic behaviors. It affects 2 to 3% of the US population, and first-line treatments include medication and psychotherapy, but 10 to 20% of OCD patients are treatment resistant. And we know that DBS, or deep brain stimulation, targeted to the ventral capsule, ventral striatum region, offers relief in 50 to 70% of refractory cases. So one of the reasons that DBS might not be 100% effective today is that DBS amplitude is constant, but symptoms are not constant. So if stimulation amplitude is set too low, you might see OCD symptoms, including distressing thoughts, ritualistic behaviors, pathological doubt, or feelings of incompleteness. But if it's set too high, you might start to see side effects of VCVS, DBS, namely hypomania and impulsivity and sleep disturbance. So during a DBS programming session, the goal is to find the stimulation amplitude that reduces symptoms without causing side effects. So it's kind of like a sweet spot. But the problem is this sweet spot sort of changes over time as symptoms and side effects evolve. So the current DBS paradigm of open loop stimulation isn't really set up to handle those fluctuations. So currently stimulation parameters are chosen during visits to the clinician's office that take place weeks to months apart. So the patient comes into the clinic, they might, the psychiatrist might turn DBS on or reprogram, and the patient might say they think they feel better, and then they go home, they experience side effects of stimulation or even new OCD symptoms. Weeks to months later, they come back and they don't feel better anymore. So tonic stimulation really fails to address the dynamic nature of OCD symptoms and side effects that might fluctuate over minutes to days in between clinical visits. So we're proposing that automatic detection of mental states related to symptoms and side effects might improve efficacy of DBS for OCD. So what that would look like is we would continuously record neural signals from the brain, use those signals to classify a disease-relevant brain state like an obsessive-compulsive symptom state, and then control stimulation parameters accordingly to relieve symptoms. So there are many, many challenges that need to be addressed in order to get here. I will talk about a few that are relevant to the paper we're talking about today. Here, the first two involve better characterization of symptom state. So there's a lack of established physical readouts of symptom state in OCD. And there's uncertainty in how symptoms vary over time at home. And the third key challenge is it is involving neural biomarker identification. So today there is no putative neural biomarker for any psychiatric disorder. And until recently, we didn't have the ability to record intracranial neural data at home. And home is really where these fluctuations in symptoms and side effects are taking place. So this third key challenge is the ability to detect neural biomarkers outside of a controlled clinical setting, and that's what we really address in this work. So getting into the paper, as you can see, this is a very long list of authors. As I mentioned in the beginning, this work is part of a large collaborative NIH UH3 effort between Baylor College of Medicine, which is where I'm doing my postdoc now, and Samir Shett's lab, the University of Pittsburgh with Jeff Cohn's group, and Brown University with David Borton's group. And the overarching goal of this project is to develop adaptive deep brain stimulation for OCD. And it really tackles the key challenges that I listed out on the last slide. Again, most notably the ability to detect neural biomarkers outside of a controlled clinical setting. So in this work, I'll talk about our first 5 study participants, who I'll refer to as P1 through P5. The first 2 were implanted with the Activa PC+S, and the second 3 were implanted with the RC+S. And so across these 5 participants, there's some overlap in principle, OCD symptoms, but the OCD symptoms manifest in different ways across each individual. All 5 participants were responders to treatment, and the Medtronic Summit RC+S rechargeability and wireless data transmission enables streaming of time-domain intracranial electrophysiology both in the clinic and at home. And this wasn't possible with the Activa PC+S system or any other current DBS platform. So therefore, in this talk and in this paper, we focused on the data gathered from the 3 participants implanted with the RC+S platform. So during DBS surgery, the DBS leads were placed in either the bed nucleus stria terminalis, the BNST, or the ventral striatum, or ventral capsule ventral striatum, VCVS, based on awake intraoperative testing. So here I'm showing P4, participant 4, as an example, where the tip of the right lead was targeted to the bed nucleus stria terminalis, which is just posterior to the anterior commissure. And the anterior commissure is the orange fiber tract highlighted on this slice. And the tip of the left lead was targeted to the ventral striatum, which is just anterior to the anterior commissure. So the stimulation and sensing contacts are indicated by the color of each of the 4 spheres, where we're sensing from a bipolar pair. Those are the green electrodes. We're stimulating in between that pair at the red electrode, and the black electrode was not used. So after surgery, the participants return to the clinic bi-weekly to monthly for DBS programming and behavioral and electrophysiological testing. So here I'm showing clinical outcomes for P4 as an example, so the same participant as on the last slide. So clinical assessments are administered at each visit, and here I'm showing a subset of the assessments over weeks since DBS on at time 0. So DBS surgery and the first DBS programming session are marked by these vertical black lines here, with DBS on again at time 0. And the Y box, which stands for the Yale-Brown Obsessive-Compulsive Scale, is the gold standard clinical assessment for assessing OCD severity. The star marks the point at which the participant became a responder to DBS at about 30 weeks. So I also want to draw your attention to the Young's Mania Rating Scale, which is used to assess hypomania, which is, again, a potential side effect of VCVS DBS. And you can see that the pink line fluctuates over time. And the other 2 scales are the Y box 2, another measure of OCD symptoms, and the HDRS, a measure of depressive symptoms, which is depression is comorbid, has a high comorbidity rate with OCD. So we have data on how this participant is doing when they come into the clinic on about a monthly basis. We know they're a responder to DBS treatment. However, a big question we need to answer in order to find a robust biomarker is, how's this participant doing in between clinical visits, and how are the symptoms changing on a daily or moment-to-moment basis in natural environments? So the what's going on in between clinical visits question is really what we're trying to get at with this work. So to get at that question, we're streaming intracranial local field potentials, both in the clinic and at home. And again, the devices used in this study allow us to record electrophysiology in the clinic and at home, so we can time sync our neural data with behavioral data. So in the clinic, we're able to estimate changes in affect in response to changing stimulation parameters during DBS programming over short timescales by estimating positive affect via facial affect recognition or automatic facial affect recognition, or AFAR. And at home, we've really made an effort to maximize the quantity and utility of our recordings by asking participants to record in a wide range of scenarios, including daily recordings, recordings during exposure and response prevention or ERP therapy, recordings during sleep, during behavioral tasks, and multi-day sprint recordings, which are essentially marathon recordings over multiple days. So participants control their own recordings at home by using a patient-facing application developed by researchers and software engineers at Brown, David Ching, Fernando Gillen, and Bradford Rohr. So participants can use this to control the start and end of their recording sessions by clicking this button after their devices are connected. They can launch behavioral tasks in this playground tab. And this app also does automatic impedance checks at the start and end of each recording, so we can check our signal quality. So each time the participants do a recording, we ask them to rate their intensity of OCD symptoms using the Roon Labs mobile application, which is tailored for OCD. In this talk, I'll be focusing on this 0 to 10 self-report slider scale of OCD symptom intensity, but the participants are also logging mood, sleep, and food intake. So to give you an idea of what the first programming session is like for these OCD participants, I'll show you a video of participant P5. So here we're recording heart rate through ECG and blood volume pulse, 64 channels of EEG, so scalp electrodes, LFP, and acceleration from RC+S along with DBS amplitude adjustments here. And so the video was used to estimate positive valence from a 0 to 5 scale shown by this pink line. And this is based on the intensity of 2 facial action units related to cheek raising and lip stretching that happens during a smile. So as you watch the video, look for positive affect to increase or decrease with smiles as DBS amplitude increases here.
You guys are shaking my body so much. My inside's not shaking. Okay, it's not shaking. How weird. It was literally when you turned it off, I didn't even realize that it went back to doing this on it. I didn't even know I was doing that. Yeah. It's not doing that right now. Oh my gosh. This is the best thing ever. Oh. Weird. It's not like I'm not internally trembling like fear-wise. Oh. Probably the biggest thing I can feel different, and so it's not like a, other than just feeling like a breeze feeling, but other than that, that's what it's wonderful. Because it feels nice and I'm not shaking, and I didn't even know I was shaking. It feels it's a good thing. I didn't realize it's like, and it like something inside felt like it was doing this. Oh, am I more anxious again? Like, why is that? Wow. Okay, guys, so freaking cool. Holy crap.
So as we just saw, this was a really emotional and happy moment for this patient and everyone in the room. This was the first time she felt relief in a really long time, and you can really see the emotion getting picked up in the data. So to take a second look at that, we're estimating positive affect intensity in response to changing stimulation parameters. And so here we saw a peak in positive affect here with a smile during the portion of the programming session when DBS amplitude was increasing from 0 to 4.5 milliamps in steps of 0.5. So in the future, we're really excited to use this data to potentially predict positive affect based on neural data. So at home, we're monitoring symptoms in between visits and over longer timescales. So these plots show the self-reported intensity of OCD symptoms that the participants are reporting through the Roon app on a scale of 0 to 10. And from top to bottom, we have P3, P4, and P5. And the colored points are these symptom intensity ratings, whereas the solid line shows the Y-box2 score. And higher Y-box2 scores mean more severe OCD. So as I mentioned, all 3 participants are responders, and the time point at which they became responders is indicated by the star here. So after their responder status is met, there's very little variation in OCD symptom intensity for P3 and P5. However, for P4, who reached responder status here at 30 weeks, you see that this participant continues to have pretty wide variation in symptom severity over days. And we thought this variation was really interesting because it shows that even after responder criteria is met, there are still good days and bad days, and symptoms are still fluctuating. And this point acts as further motivation for developing an adaptive system that might be able to respond to these fluctuations in symptoms and prevent this variation from happening. So each time the participants do a recording, we ask them to report their intensity of OCD symptoms, and we can associate each recording with a particular symptom level. So this bar chart summarizes the amount of data we've collected at each symptom level, along with how much data we've collected during behavioral tasks, during exposure and response prevention therapy, and in total. So in total, we've collected over 1,000 hours of LFP across these 3 participants. And notice that the majority of LFP data is associated with symptom intensities below 3 out of 10. So we've generally noticed that the participants are more likely to engage in recordings when they're feeling well, which is kind of a reporting bias that we've noticed. So we're also doing multi-day sprint recordings. So here I'm showing an example sprint recording where participant P4 recorded for over 30 consecutive hours with breaks for recharging. So the participant recorded heart rate and acceleration on the Apple Watch, and LFP and acceleration on the RC+S, and submitted OCD intensity readings on about an hourly basis. So the 90 seconds of data that I'm showing here just shows an example of each of these streams. So P4 did one 3-day sprint recording where there were natural exposures to OCD triggers on days 1 and 2. And this recording stood out as a really great opportunity for analysis because we don't often see such wide fluctuations in logged OCD symptoms within the same recording over such a long time span of 3 days. And since our goal is to develop a classifier that distinguishes between low symptoms and high symptoms, in order to do that, we need to collect a lot of data associated with both states. So LFP availability over the 80 hours here is shown by the orange shading, and self-reported intensity of OCD symptoms is on the y-axis. And here we see a range from about 0 to 8. So to analyze the LFP data, I computed spectral power and frequency bands of interest for each 2-minute segment of data around each symptom intensity rating. So if there's a change in neural activity in the ventral striatum during symptom provocations, natural symptom provocations, we might be able to pick that up in the spectral power of LFP. So here I'm showing results from that analysis where the y-axis is spectral power, x-axis is symptom intensity. So we're looking at delta 0 to 4 hertz, beta 4 to 8 hertz, alpha 8 to 15 hertz, beta 15 to 30 hertz, and gamma 30 to 55 hertz band activity for left in the first row and right in the second row VCVS. And we found that VCVS spectral power shows some correlations with OCD symptom intensity, where the correlation between symptoms and delta band power showed the strongest correlation, and theta, alpha, beta, and gamma band power showed relatively weaker correlations. So another opportunity to record neural data during OCD exposures and variable symptom intensities is during exposure and response prevention therapy, or ERP. And all of this all of these therapy sessions were done via telehealth due to COVID. So the exposure in ERP refers to exposing yourself to the thoughts, images, objects, and situations that make you anxious or start your obsessions. Whereas the response prevention part of ERP refers to making a choice not to do a compulsive behavior once the anxiety or obsessions have been triggered. And all of this is done under the guidance of a therapist. So during the 2-month cognitive behavioral therapy booster phase of our study, participants do 13 ERP sessions, and here the chart shows data available for each stream again during each session over the 2 months. So I boxed out this particular session that I'll show you a video clip from on the next slide. During this session, the fear trigger was a wart on the hand, so this photo here, and this plot is showing heart rate collected over the whole session, and throughout the session, the participant is asked to verbally rate their subjective units of distress, or SUDS rating. So the video clip I'll show on the next slide corresponds to this gray shaded area here right after the trigger was introduced. And again, this is all done over Zoom with the participant at home and the clinician at the office. So the participant in this video has very, very severe OCD specific to contamination, and the thought of having a wart was very distressing for him. And the image of the wart helps to facilitate thoughts about having a wart. And he identified thinking about warts as a rather difficult exposure, and he had to complete a number of different contamination-related exposures to even build up to thinking about doing this. So he worked his way through this hierarchy, and after enough practice, thinking about and seeing images of a wart was within his ability to tolerate. So this was one of, I think, the last ERP sessions in the 2-month phase.
I'm recording on the watch as well.
Can you see this okay?
Yeah.
So right here is where we're talking.
Yeah, I see it.
0 to 10?
Yeah. 8.5. Oh gosh. And now I'm looking at my own hands.
That's good.
For one of those.
So your OCD is concerned that you could have one right now?
Yeah.
Okay. This is really good work. I know that this was a huge trigger.
Yeah.
Even that you're willing to look at this, to purposely look at this, like that's awesome. Let's keep rolling with this. So what are we doing?
Okay.
Why is this wrong and dangerous? How are we just gonna resist those urges? We're not correcting this.
Yeah. Okay. You can still see me, right?
No.
Yeah. Okay.
Yeah, I can.
That's 8.5. Still very strong 8.5. Oh god.
So overall, the ERP sessions across all 3 participants, we really did see a high range of SUDS or subjective units of distress. And so for this reason, the LFP recordings collected during ERP teletherapy really present a unique opportunity to capture neural data during fluctuations in symptom severity. So as an example, I'll show results from analysis similar to the analysis of the sprint recording I already showed from this recording boxed out here. So here the SUDS ratings range from 3 to 7.5 throughout the ERP session. And the vertical black lines show the times of each exposure throughout the session. And as you can see, the time course of the SUDS ratings show some habituation after each exposure. And that's really the goal of ERP therapy. So I analyzed the LFP data in the same way as I did before. by computing spectral power and frequency bands of interest around each 2-minute segment, around each 2-minute segment of data around each symptom intensity rating. And here you can also see some interesting correlations between SUDS and spectral power across frequency bands. And while delta band power did not show the strongest correlation here across all of the bands, we did see that the relationship between delta band power and symptoms was preserved across planned exposures during ERP and natural exposures during the sprint recording that I showed from P4. So the recording platform I've described enables this type of analysis, and future work will determine the degree of consistency of these brain behavior relationships across patients and their potential as a biomarker for ADBS applications. So to conclude, for the first time in patients with a psychiatric disorder, we have demonstrated the utility of recordings collected at home, which vastly increased the quantity and ecological validity of the recorded data. We captured over 1,000 hours of LFP data at home across various symptom levels and 2 modalities of treatment, ERP and DBS. We demonstrate the ability to measure VCVS spectral power during natural OCD symptom provocations at home. And we identified low delta band power as a candidate biomarker of high intensity of OCD symptoms, which could potentially be used in an adaptive DBS system in the future. So continued opportunities for long-term naturalistic intracranial EFIS recordings will propel biomarkers discovery for OCD and other psychiatric disorders. So next, we hope to use the wealth of data that we've already collected both at home and in the clinic to identify neural biomarkers of both OCD symptoms and side effects of ventral striatum DBS. Some other things that we're doing that I mentioned briefly in the clinic is we're doing behavioral tasks such as a provocation task to elicit OCD-related distress. And we can also use the ERP recordings we've already collected to do that in a more natural setting, where at home we can really have participants interact with their OCD triggers that are kind of holding them up in their real lives instead of, you know, maybe something more fake on a computer screen or representative on a computer screen. We can capture both anxiogenic and anxiolytic responses to programming in the clinic. And we can have participants do long recordings at home, rating their OCD symptom intensity throughout the recordings while they're just living their daily lives. And so together, all this data will hopefully lead to biomarker identification and development of an ADBS system for OCD. So next, I want to talk about the emerging challenges that I think the field faces next. So we really need to identify robust behavioral markers. The utility of self-report assessments are limited, I think, as they rely on patient insight into their own psychological state, which is variable, and they aren't continuous. Like, you can't ask a participant or someone how they're doing at every moment of the day. So I think we need to develop enhanced passive behavioral state monitoring. For example, we could use wearables that track exercise and activity level, and that might provide more insight into measures of symptom state for psychiatric disorders. And I think this would help reduce the burden on patients as well. So next, we need to automate electrophysiological data collection. So more than half of the LFP data collected for participant P4 was associated with symptom intensity ratings of below 2 out of 10. But as you can see from like all the ratings that he gave us throughout the study, many of them were associated with higher symptom levels. So he was more likely to do recordings when he was feeling well. So we need to decrease the burden placed on the participants and automate data collection. until LFP recordings can be triggered without participant input, I think they'll be subject to the participant's motivation, anxiety, and mood. And lastly, we need to harness network connectivity estimates. OCD and obsessive-compulsive behaviors implicate symptomatic brain networks. So we need to increase the coverage of our approach to a few key regions of the network, such as the orbitofrontal cortex or anterior cingulate cortex. By increasing the coverage, our ability to identify a biomarker and design an ADBS system would improve, leading to hopefully better outcomes for severe cases of OCD. So in the next phase of our UH3 study, we're doing just that. And we've already implanted 2 patients so far with orbitofrontal ECOG leads, as well as the ventral capsule ventral striatum leads. The orbitofrontal cortex, or the OFC, was chosen as our cortical target due to seminal work using imaging techniques that have found that overactive connectivity between the cortex and the basal ganglia is a signature of OCD. And this hypothesis is also supported by characterization of functional pathways. So in phase 2 of the study, we're looking forward to using connectivity estimates to see how, for example, how coherence changes between the OFC and ventral striatum over time or with symptom provocation. So with that, I'd like to wrap up and acknowledge my PI, Samir Sheth, my thesis advisor, David Borton, and the PI of the study, Wayne Goodman. And I want to thank our collaborators at the University of Pittsburgh, Jeff Cohn's group, for their computer vision estimates of facial affect. And I would also like to thank the Open Mind Consortium for all the support throughout data collection and data analysis. And with that, I would love to take any questions. Thank you so much for your attention.
Thank you, Nicole. Great team and fantastic work. I mean, the results are highly novel, and I believe this paper can become a major one in the closed-loop DBS field and, you know, new clinical avenues for sure. And thank you, everyone, for your interest and attention. I guess we can take the questions now. We have a few. All right. The first question is, did you capture autonomic data from the Apple Watch? If so, did you find any correlation between something like heart rate variability or heart rate and the symptoms, SUD scores? Really cool work. Also, did you notice that DBS stimulation altered the delta power SUD's correlation you noted? By the way, the comment came from Nicholas Trapp.
Thank you so much for the question. I might ask you to repeat that second question there because I was still thinking about the first. So the first question was about autonomic measures from the Apple Watch and whether we correlated that with like symptom provocations or symptoms. Yeah, so that is something that I'm really interested in. Unfortunately, the app that we were using was not able to capture heart rate variability, but instead captured just heart rate kind of averaged over like 10 or 20 second intervals. So at first glance, I looked at a couple of sessions and did not see like any correlation between symptoms and heart rate, but I would be more optimistic about something like heart rate variability based on like the literature on that, like as a metric related to like anxiety or arousal. So yeah, I think that's a great idea and something that we would love to get in the future. And can you remind me what the second question was? Yes,
sure. The second part is, did you notice that DBS stimulation altered the delta power SUD's correlation you noted?
Oh, yeah. Well, yeah, that's something that I looked at. And we did find that there was an increase in delta band power over weeks since DBS on, but that increase I think was confounded by changing impedance values over time, and I think it ended up being not significant. There's other ways that we could look at it though. For example, by doing an experiment where we vary dBs amplitude, for example, like within the same session, to try to create like a dose response curve of delta band power versus like dBs amplitude. That's something that we're actually looking to do. Well, we're working on it right now. So I'm kind of hoping to see that we're able to, you know, see that dBs, increased dBs amplitude increases delta band power and like also related behaviors like feeling better. Yeah, but we haven't done that yet.
Thank you. The next question is coming from Duvan Hoffman. Are imbalances in the communication between right and left hemispheres used as a biomarker?
That's also a really good question. I have not looked at coherence between right and left hemisphere, like VCVS yet. I think that's a really good idea.
He also a comment just related to the question. In auricular medicine, a field of neuroauricular, sorry if I'm mispronouncing it, neuroauricular modulation, we can identify this via laterality disturbance, which is often associated with OCD, HDHD, and related issues.
Yeah, that's really helpful. Thanks.
Okay, thank you. I have also a few questions. Like, I mean, obviously you've been recording data for weeks, and can you share your experiences with, you know, working with big data sets? And there are multi aspects of the whole, you know, database, like EEGs, LFPs, behavioral data. Can you share your experiences? How you know,
yeah,
go through this?
that was like probably the biggest challenge of my grad work, to be honest, like wrangling all of this data, including, like, as you say, the EEG, the video, synchronizing the video is really tough with the neural recordings. Audio, you know, we have all these different inputs, it's a lot of data, and it all has to be synchronized. We did that, most of the synchronization as a post-processing step. With the DBS patients, as you all know, for the PC plus S, the data was stored on board the INS, whereas for the RC plus S, it was streamed out to a computer. And it was important that that computer wasn't doing anything else besides capturing that data to reduce packet loss, which also created a lot of like processing issues because like the RC plus S streams the data out, and you know, you lose data packets. So that creates challenges for doing any analysis that's like continuous in time. Like, how do you handle you know, doing coherence between two signals when you have packet loss in different places throughout each of those time domain signals. So dealing with packet loss was really challenging. And then this is, I guess, unrelated to like the amount of data that we were collecting, but another huge challenge was dealing with DBS artifact removal.
I was going to ask, I mean, it's a big challenge in the field, right?
Yeah. Because you know, there's always the confound of like, is the signal we're looking at an artifact, or is it real? So that's like the question I ask myself every day and make myself crazy over. So there's a lot of things that we thought about to be confident in these results. And yeah, another huge challenge I think with this data set was like, we're distributing tablets to participants at their homes, and the participants are all over the place. I was in Providence, Rhode Island. The clinical side was in Houston, Texas, which is where I am now for my postdoc. So like, kind of engaging in the collaboration effectively while leading data collection was really tough. And like making sure all of the data uploads happened, like setting up automatic data uploads and stuff like that was essential for like being able to do analysis. Yeah, so it was a huge challenge. Thanks for that question.
Well, you did a great job, apparently. Everything looks great. I think we have one more question coming from Martin Fish. LFP characteristics are expected to differ with variance in patient recordings, location, lead target. What is your experience so far?
LFP, wait, what was the first two words? LFP what?
LFP characteristics are expected to differ with variance in patient recordings, location, lead target. So what's your experience?
Yeah, so like another big question that I haven't really touched on even is where exactly are the recording electrodes? Our scans indicate that the deepest contact on each patient, for each patient, is seated in the gray matter target, whether it's the VNST or VCDS. Whereas as you kind of climb that electrode shaft, you have the deepest contact, which is the deepest recording electrode, stimulating contact, and then the next one up is the second in that bipolar pair, other recording contact. So that recording contact, I think, is often in the white matter. So it's kind of like, you know, what exactly are we recording from when one of the contacts in the bipolar pair is in gray matter and the other is in white? We have noticed across participants, and this wasn't something that we put in this paper, but an interesting thing that we've noticed is we've seen some high amplitude events that sort of look like spikes in LFP in the ventral striatum activity, or ventral striatum and BNST activity, that, you know, is kind of uncharacteristic of other regions that people record from in, you know, with DBS electrodes like the STN. So that's one thing that we're kind of looking into right now. Yeah, I hope that answers your question.
Thank you. It may not be the scope of the paper, but I was wondering, have you recorded intraoperative LFP and look at the signatures or how they change over time with your chronic recordings?
Have I sorry, what was the beginning of that question?
Did you record intraoperative local field potentials? I mean, any recordings during the surgery, and if so, did the characteristics changed over time? You know, with respect to your chronic recordings?
Yeah, so that's actually something that I just recently started doing now that I'm at Baylor College of Medicine, which I'm really excited about. Yeah, we're recording during awake testing during the surgeries. And so this is how the neurosurgery team decides whether to place the stimulating lead in the ventral striatum versus VNST. They turn on stimulation during the surgery, ask the patient how they're feeling, and then if they don't get a desired behavioral response, like increased positive affect or mood, energy, then they'll actually move the lead and see if the other target produces that response. So I'm really interested to see, you know, can we look at the intra-op recordings and make connections between which target gave us the desired and contact gave us the desired behavioral response. And I'm interested to compare those recordings, you know, with a kind of more robust back end to the recordings we get on board the RC+S. Because in the OR, we can record from all the electrodes on the lead. And with our new cases, we can also record from like the ECOG electrodes. So I'm excited that we're now getting that data. Yeah.
Cool. We have a new question from Michael Saffron. Do you think segmented DBS lead could benefit your research or just make it more complicated?
I think the answer is both. segmented leads give us, a higher degree of freedom, obviously, when choosing these, like when optimizing simulation parameters. Something that I've been thinking about is like, well, if we want to compare two different targets, let's say after the electrode is placed, can we simulate on one side of the lead and capture and simulate what would be the ventral striatum target, and then can we simulate on the other side of the lead and simulate the BNST target and see, which one is better, long term, instead of just relying on this acute behavioral response for lead placement. Yeah, but it's when you think about if you want to design an experiment to test all of the different possible simulation parameters, then you have current steering as an option, where there's a million different configurations or many different configurations. That's a lot of time that the experiment would take. So that makes things kind of difficult in terms of experimental design, but that's definitely something I'm thinking about in the future, like in our depression trial that's going on, especially.
Thanks so much, Nicole. I don't think we have any other questions. Yeah, I think we can, close up the session. Thanks so much for the nice talk and nice comments. And see you all in next month's Neuromodulation Club.
Thank you so much for having me, and thanks everyone for your questions and attention.
Bye.